uh, who is professor of systems biology at Imperial College London. His research focuses on the use of physics approaches to provide quantitative understanding of cellular processes, mostly in sensing and signaling. And the group's aim is to extend the success of physics to living matter and to unravel the underlying design principles hidden within the enormous biological complexity. Today, Professor Endres will talk about searching for robust Turing patterns through evolution and AI. And this seminar is recorded and will be uploaded to our DCB Turing website, where you can see this talk and also our previous talks and recordings. So let's welcome Professor Andres, and I will start recording from now. Yeah.
Thank you very much to the organizers for having me and for the platform here as well to present, and also to everyone for attending. So good afternoon. I have now on the right side the thumbnail pictures of attendees. Is it in the way, or do you even see that? Is that okay? Okay, so I guess it's fine. Okay, so anyway. Okay, so what I'm going to talk about today is Turing patterns. So that's work over the last few years. In particular, part of the work is done with a synthetic biologist at Imperial College, Mark Iserlan. And then I will sort of second half, I will switch. So we do mathematical modeling for that. And the second half, I will switch to more recent work, which started as undergraduate projects. And the work is still unpublished. So that's less polished, basically. So I don't think I need a sort of full hour. So in case there are any questions in between, do feel free to interrupt. And if that's possible, then that's totally fine. Just maybe one comment, evolution here in the title. So I'm not going to talk a lot about evolution. It's more at a conceptual level. So I'm not specifically modeling how Turing patterns might provide some evolutionary advantage, but the search through parameter space and model space might be relevant for evolution. Okay, so here's a bit of an outline. So at first, I wanted to spend a bit of time recapping the Turing mechanism in case some of you or students might not know, and in particular, sort of highlights the connection to biology, and in particular, that robustness issue is an important problem, and I will define what this means. So we'll start with the synthetic biology approach in our modeling, and as I said, in the second half, it's about how evolution or whatever, I mean, how that works, how to discover these parameter spaces which lead to Turing patterns. And the first part of that section is about limit cycles to use these. And then in the last part, it's about neural networks, how to discover Turing models and ideally ones which are robust. Okay, so as theorists, I would say in particular, we are very much interested in discovering structures and patterns in nature. And so here one picture which has various different structures. So the first one here at the top is a bird flock. So we're all very familiar with that. So here, that's a very quite well-defined problem because we might get away with certain reasonable modeling assumptions like pairwise interactions within a certain radius or topologically speaking. We might not care about the individual properties of each bird, what it had for breakfast and so on. So we can glance over some of these details. But then if you go to so different scales, for instance, a zebra, which is a classic problem which people describe in terms of Turing patterns. So the connection to Turing is less clear because here we have a sort of a macroscopic object like a zebra, and the mechanisms which play an important role in Turing don't play so well at these length scales. But nevertheless, the idea is here is that whatever these pre-patterns which form, they happen much earlier during embryogenesis, and that might well be a Turing mechanism, actually. So then we can go further down in scale, and then we can talk about microbial communities, division of labor, and so on in biofilms, and there are certain structures. And this might be a nice testing ground for various theories and sort of experimentation as well. Because I would argue it's a colony level, so we know a lot about the processes. There's physics, obviously, for instance, diffusion, but then also the chemical reactions we understand in principle very well. in terms of gene regulation, for instance, and how it relates to cell growth. But also some things are controllable in experiments, not everything. And so there are some challenges, of course, as well. And in terms of mechanism, I mean, especially as a theorist, you can think of different ways how to approach this. I mean, the physics approach might be more phase transitions. Then the classic approaches might involve things like the French flag. I will briefly discuss this afterwards. And of course, Turing mechanisms. Yeah, here's a picture of the famous paper by Alan Turing from 1952. So it's quite a remarkable paper because he describes a mechanism for pattern formation, spatial pattern formation, which is somewhat counterintuitive. So first of all, he has these morphogens like 2, an activator inhibitor, and by themselves, the chemical reactions don't do anything interesting. They lead to a homogeneous steady state, like flat concentration in space, nothing interesting. But then you turn on diffusion, and all of a sudden, the thing becomes unstable. And that's counterintuitive because the reactions themselves and diffusion by itself would lead to an even distribution of molecules, and that's nothing interesting, but together they form this instability. So he talks about 6 different outcomes. The stationary pattern is one of them. It's an interesting one here. And then also in a quite modern way, he talks about biological applications. So these are underlined in green. And also quite remarkable, very early on in the 1950s already, he talks about the challenges of doing multidisciplinary research. Said it's challenging for the audience to understand, the mathematics and biology and chemistry. So that was quite ahead of its time. Okay, yeah. Here's one of the pictures from the paper, which I find interesting. So he does numerics as well, and it's sort of calculated by hand. He also alludes to a future paper on using computers. Yeah, I guess that was never written, this paper. Yeah, so here's a little bit of a recap of what this mechanism really entails and how it might be different from other mechanisms. So, it's quite different from the traditional, at least equilibrium phase transition. This is clearly non-equilibrium. We generally don't talk about the thermodynamic limit. We have a finite domain, like an embryo, for instance, and we don't have mass conservations. So molecules are produced or degraded. And so in this context, we would talk about a bifurcation. The simplest model, which Turing introduced, is a 2-species model, 2-morphogen model, U and V. And they are described by this reaction diffusion equation. I mean, Turing started with a discrete model. And so you have reaction terms, and then you have the diffusion terms, the Laplacian here, second derivatives. And generally, what you often do, and here's not really specified the reactions, so often you draw these diagrams, which is shown here, and hope it's not covered up now by the little thumbnails. I guess I can move it. So the activator activates itself and activates the inhibitor, and vice versa the inhibitor. So that's one type of topology, how you wire it up. There's another one, which is called the substrate depletion one, where the activator actually basically inhibits the inhibitor and so on, but it self-activates. And it has it's an out-of-phase pattern, this is in-phase. Anyway, so now we can go to the phase portrait. So I mentioned already in this concentration space of the U and V concentration of activator inhibitor, without diffusion, that would be stable. So things flow towards a steady state. And in terms of space, that would be, flatly distributed, nothing interesting. But then this distribution, that would become unstable, and there would be a wave number dependent pattern forming in space, which is shown on the right. And that would be describing the initial instability and so on. Okay, so here basically is a picture of the 6 different outcomes, which Alan Turing already described. There can be also oscillations or traveling wave. For traveling wave, that's certainly very much boundary condition dependent or initial condition dependent. And then the stationary pattern we are mostly interested in right now. So the first thing one does, and Alan Turing did this, so you study the behavior around the stable steady state when you introduce diffusion by linear stability analysis, and then you study these eigenvalues and so on of the Jacobian. But anyway, the eigenvalues are now wave number dependent, and the idea is now one of these 2 eigenvalues has to become positive for the thing to become unstable as a system, and then you have these perturbations at a particular wavelength here, which grow. But initially, for a k equals 0, that's means no diffusion. Z has to be stable, so negative. Yeah. Okay. So now what is the issues with the Turing mechanism or the Turing models? So a later model, which is a French flag model by Wolpert, was much more popular among biologists. It was less technical. So that describes a morphogen gradient and how it's read out. And that provides positional information. So for each position in the gradient, you have a concentration which can activate gene regulation, for instance, and can form patterns. So this is about readout, not about the emergent symmetry breaking, which you have in Turing, which forms patterns from scratch. It's very different. But it has applications to biology. Think of Bicord gradient and Drosophila embryogenesis. That has nice applications. With Turing, the relevance to biology is somewhat complicated. So there's some nice example where Turing certainly is highly relevant. A good example is the digit formation, meaning finger formation in mice, and then the fish skin pigmentation in zebrafish. So for instance here, so each of these models is essentially a two-equation model or three-equation model. So very simple. So the complicated developmental biology is essentially put into these rate constants, but it's surprisingly predictive. So for instance, it can describe a mutant, which is called the polydactyly mutant, which leads to too many fingers. However, that is still more complicated because it also needs morphogen gradients to align the fingers, for instance, in the radial direction and not, let's say, perpendicular. So that leads to further stability and reproducibility. In terms of fish skin pigmentation, the models don't even have the usual diffusion. They would describe how pigment cells do random walk and interact and so on. So again, a very simple model, but surprisingly effective. So what I mentioned at the bottom here is a classic robustness issue, and that's one of the downsides. So Turing patterns are very simple, and it doesn't look really like biology, and you have to fine tune. So so this is sort of summarized here. So that was actually a paper from my collaborator with Michael Stumpf, another theorist, and they systematically searched through all two-node like Turing and three-node networks where you have one of the nodes non-diffusing. So by exact enumeration, looking through all the networks and basically asking, first of all, with more nodes, does it become more robust, meaning the larger fraction of parameter space leads to a Turing instability. So larger networks seem to generally form more robust Turing patterns. But the important thing is also that the majority of these networks, these topologies, however you wire them up, lead to a Turing pattern, but the parameter space is tiny, like a fraction of a percent. And so that is a classic fine-tuning problem in Turing. Of course, it's not the full story also. There's another nice paper here by Diego, and this is basically what I'm saying is a statistical property on average, but there are certain topologies which are much more robust than other ones. So that's a nice paper which delves into that. Okay, so the question now is, how to link this to biology? Developmental biology is super complicated. These networks are super simple, so there's some misalignment already. So another route would be to now turn to synthetic biology and to build these from scratch to see, if you can understand it by building. And generally, since synthetic biologists often quotes this Feynman mantra, what I cannot create, I do not understand. So that's, of course, a motivation to understand it by doing so. Okay, so there have been some synthetic biology efforts before, and maybe two are noteworthy to mention here in particular. So one is, so both of them try to implement this very simple two-node, two-morphogen classic model. This one on the left is done in bacteria, and the other one here on the right is an eukaryotic system, a mammalian system. And but in both cases, the bottom line is that these are not really Turing patterns. Also, there are some patterns. So one is called stochastic Turing patterns. So the thing is, they connected nicely with their model, arguing that the power law, there's some power law in the power spectrum and so on. So there's a nice match between model and experiment. But they sort of admit in their theory sections that the model doesn't describe Turing. And the reason is that the diffusion constants are too similar. So one of these Turing conditions is, the mathematical conditions is that the diffusion constants have to be sufficiently different, at least for the two-node network. So maybe I should mention the Turing conditions and why it's so fragile is with the bottom line is um, that there's these conditions, it has to be stable without diffusion and unstable with diffusion. And these are mathematical conditions which constrain the parameter space. Yeah. And so here we have this problem that diffusion of small molecule is too similar and that condition is not fulfilled. Similar on the right side, so they call it a solitary pattern. It's basically a bistable, excitable system and also not a classic Turing. Okay. Okay, so that was then the motivation for my collaborator Mark Isalan in his lab, and in particular postdoc Yuri Tika, to now take one of the most robust networks, 3-node, and implement this in actual cells. And this is basically an equalized cell. And now each of these 3 nodes is divided up into 2 genes. And the reason is that basically the activation and inhibition functions are separated onto 2 different proteins. So we have 6 genes. And yeah, so to read this out, basically there are fluorescent proteins as well downstream of these two, RFP and GFP. We have small diffusible molecules like these are from quorum sensing, similar to one of these other papers I showed you on the previous slide. PC and OC14, they are also secreted and then sensed by other cells, and that's how they communicate. And what else? Then there's these external knobs to have some controllability once it's implemented. So ATC inhibits that loop or that inhibition, and IPTG is that one here. So there's a way to tune it as well. And these are sitting on plasmids, these different genes. Yeah. So okay, and then so here's a corresponding model from my PhD student Martina. So she implemented these 6 genes by 6 equations, reaction diffusion equations. So here you see these so-called Hill functions, which are factorized. So these describe gene regulation, in particular non-competitive binding to the promoters. And then these have an effective diffusion because they're regulated by these small diffusible molecules. And so what she does is essentially she did these cellular automata simulations, so describing the cell growth, the colony, and then solving wherever cells are these reaction diffusion models. And outside, you just have degradation and diffusion. So the problem now here is that's a nice model, but it has, I think, over 30 parameters. And so, what do you do with a complicated model like that? Yeah. But okay, let's talk about so what is the main issue here? Yeah. So the good news is, in experiments, early on, the experimental lab produced these really nice patterns. You see in the top row, which we described in different ways or different names here. But the issue is that they're more or less under very similar conditions produced. And so you see the variability is huge. So there's a problem which is already reminiscent of Turing, the fine-tuning requirements. It's hard to control. On the modeling side, you see at the bottom here simulations, and you see they more or less seem to match. But here's a caveat is that this is based on a global parameter search with Latin hypercube sampling, searching this large dimensional space quite efficiently, but then essentially picking parameter combination which makes the simulation look like the experiment. This is essentially fit by eye. And these parameters are vastly different, yeah, and some of them might not even be sort of relevant biologically speaking. So here you see already these issues. Nevertheless, the model can be useful. So first of all, you do linear stability analysis and you can see the different outcomes again. Turing 1, which is a classical Turing spatial pattern, is very small percentage. So you see already, Hopf is oscillations, they're much more predominant and so on. And what you can also learn is that to get Turing, you need to have these so-called interactions between the species. They have to be matched, meaning they can't be in the saturated regime where they become very much independent of the concentration of the inputs. So that's not desired. So you can learn this as well. Another outcome from the modeling was that increasing ATCs, this external molecule, should be cranked up in concentration to have a higher chance to discover Turing. Maybe one other thing to point out, linear stability analysis describes the initial onset of pattern formation. It's normally done on an infinite domain. It's not in the sort of realistic scenario. But when you simulate it realistically, like the growing colony, the outcome can be very different from what you predicted, let's say, from linear stability analysis. Even so, these cases are not these exceptions. But the point is, in this nonlinear system, you have multi-stability, and if you're kicked out from one state because of the Turing instability, you might end up in another stable state, and the Turing pattern has disappeared. Okay, that's just to mention. Okay, so what do we do with this huge many parameter model? That reminds you of this von Neumann elephant. With too many parameters, you overfit and you can describe anything, including an elephant. And so what do? First of all, you do non-dimensionalization, which is sort of illustrated here with a simple two-dimensional model. So first of all, you do some parameters. So you reduce the numbers of parameters. And second, which is a cool byproduct, depending how you do it, you can get ratios of concentrations. And so as effective concentrations, so unitless. And so the point is now that because experiments have, let's say, fluorescent intensity, you don't have concentrations anyway, and these ratios are more useful for describing the data. And of course, some parameters are determined experimentally, like diffusion constants. And so another thing we did here is we didn't fit to the pattern directly, which would be the ideal scenario. So what the experimentalists did, they created these subsystems, remove certain parts from the complicated network, and then look at these in solutions, these cells, so without pattern formation, describe input-output curves of this reduced model, the reduced system, and then we can describe these dose response curves more easily and fit them with a reduced model and get some of the parameters that way. So that is basically illustrated here. So here are some two submodels being fitted. And the good thing is, once you have a fit and some information about, what your desired confidence interval is, and here's that's a covariance matrix. So the point is you can estimate confidence intervals on your fits. This is shown by these lines. So you can sample around the best fit from within these confidence intervals. And now you can ask, is Turing more enriched, or among these Turing patterns you might see here, does it look like the experiment, and so on. And that's essentially what we did. So here's, let's say, a summary from the paper. So now, they turned out then ultimately more reproducible. So here are a few examples of similar stripe patterns. Of course, if it's Turing, it should be the peaks in intensity, going along here should be equidistant, so there should be a well-defined wavelength, which is sort of illustrated here. The model predicted it's out of phase, so activator high, inhibitor low, so that's somewhat reproduced here. And Turing would predict this thing is not scale invariant, so a larger colony should have more rings, but not, a larger wavelength. So that's also fulfilled. And there are some other things, sensitivity to boundary conditions and so on. So there are some things which sort of allude that this might be Turing. Here's a time-lapse movie on the left from experiments, and on the right, there should be a simulation. Okay, so of course, I mentioned, the sort of scenarios to overcome the question of, how to fit actually this complicated model, by looking at the subsystems and so on. But, ideally, of course, you do some parameter inference, you take directly the spatial pattern and now take your model, which you implemented in synthetic biology, and try to infer the parameters. And so we haven't in the end succeeded to do the experimental patterns in the parameter inference, but we worked on this as well, the inference problem. And this was work done by my former PhD student, Antonio. And I want to just briefly talk about this very briefly, because this is already also published. So the idea is, to prove that it's Turing, ideally, we take a model and show for some parameter combination it can produce Turing. That's the so-called inverse problem. So input is a pattern in the model. You can test the approach, whatever you're doing here, using numerical patterns, first of all, and try to infer. You can corrupt them by noise as well. Or ideally, of course, you take experimental patterns. So we did do experimental patterns, but from chemical Turing patterns, which are much more reproducible and less noisy and so on. And so what Antonio did, he compared two methods to do the inference. The first one is least squares, what I already described on the previous approach with the submodels. So least squares describes, you maximize the likelihood to observe the data given some parameters. And so that's the classical fitting. That is surprisingly simple, but hasn't been applied to Turing. It was thought as an ill-posed problem. For a pattern, you can get multiple parameter combinations or multiple solutions. But if you get one, we're already quite happy and we can explore locally. And then the other one, the main thing we wanted to do actually is the physics-informed neural networks, PINs. So here what you do is essentially for each pixel X and Y coordinate here, you have a large network and you approximate the pattern you want with this complicated function here, using essentially the universal approximation theorem. And then you can, of course, fit with many, many parameters here this pattern. But the question is, does it sort of relevant, this pattern? I mean, so we can constrain it more by calculating also the derivatives on these fields. And then you can include it in the loss function terms, which describe also the requirements that you want also this pattern to fulfill the Turing PDE equations. And so doing so, you can get the parameters of the model, because this is great, because here you have, let's say, thousands of parameters, and then having a few more to fit is not a big problem. So that's remarkably good in terms of scaling with parameters, numbers of parameters. You don't need a lot of data. You technically just need an initial image. So it's also unlike normal deep learning methods where you need lots of data. That's a really cool method, actually. And so here, very briefly, just sort of the main output here. So we are comparing these squares and the pins. The general idea is here to take some numerical pattern on the left and then to corrupt it with noise. These are the noise levels, additive noise, very simple. Here you can't see the noise because the noise is tiny, less than 1%. So the idea is now to take these noisy images, to do the inference, to get the parameters, and then to do the forward problem, solve with these parameters a pattern and see if the pattern looks like the original pattern. And here you see with less than 1% of noise, it already fails. So here you don't get the spot pattern anymore. It's very sensitive, even so it can be rescued with more data. The pins, on the other hand, you can corrupt with much more noise. Here you can actually see the noise between 15 and 20%, then it fails. And so how do we look at the performance? This is just anecdotal here. We look at the relative error in parameters, how well the parameters match. And then also what we do is we look at the errors in pattern directly, but not pixel-wise. We look at the mean squared error of the radial average power spectrum. So you Fourier transform effectively and you get the main features like wavelengths and so on. And it's independent of if the spots start moving around. It's independent, invariant to these kind of details. Okay, so here's the main difference, so least squares. So using three different Turing models as a function of relative added noise. So the more the added noise, the less good the parameters are. Yeah, that makes sense. And it's going in a smooth curve. When you look at the pattern with additive noise, so they go up in steps, meaning they sort of break in discontinuously because wavelengths changes, spots move around, and so on. So that's also somewhat understandable. With the pins, it's very different. So the errors are huge as a function of additive noise. So these errors are somewhere up here. And but interestingly, the patterns are much more robust. So as I said, they break around 15% of additive noise. So worse in terms of parameters, but better in terms of robustness and resilience. Okay, very briefly then, Antonio applied this then to a classic chemical reaction system, which forms Turing. This is photoactivated. So it's basically a light cone expanding and allowing the pattern to form. And the good thing is here's also a classic model which supposedly describes this pattern quite well. This is this one here. And so as before, we corrupted the numerical solution of that and did it in also the experimental one. The problem is here, what you see is some sort of spectroscopic measurement. You don't really, you don't know what is the activate inhibitor. So we made certain assumptions. We have certain scaling variables as well, scaling parameters. So you have additional parameters to fit. It works well for the numerical thing as before. For the experimental one, it predicted some spots, but the spots seem to be emerging anyway in the experiment. So we are quite close here. Okay, so now I'm coming to these two sort of ongoing projects, which started as undergraduate projects. And so this has to do now with this whole robustness issue I talked about. So So this basically, this one here addresses now how to find these small Turing islands in parameter space. You know, first of all, how does evolution find these? And then also, of course, there's also noise in systems. And so how do you make this robust? And so the ideas here is that a lot of things in nature, basically everything is coupled to limit cycles, to cycles like cell cycle, obviously circadian clocks, light, night, day cycles. So many other examples. And whenever you have negative feedbacks and delays, whenever you have delays, you get instability in oscillations. So here's three examples from a review paper. And the classic one is, the repressor, one of the first successes of synthetic biology, 25 years ago or so. Yeah, 25 years. So three repressors mutually repressing each other. And the thing is already, here you see the fact that you could build this very early on with synthetic biology and oscillator, but Turing patterns much later. So you see already oscillators are much more easy to implement and more predominant in nature. And so the idea is now, and that's where evolution comes in, that this might aid the finding of Turing islands in parameter space. So the idea is, go back a couple billion years ago. So you have a clump of cells now, and each one has some a different, let's say, concentration level slightly and so on, but they're weakly coupled by diffusing molecules and so on. And now, you have these, when you look at this combined parameter space, this high dimensional one, some of these clumps of cells now might sit here outside Turing islands, most of them, let's say, but then rarely, they hit a Turing island, these clumps of cells. And so maybe they have a small advantage over other ones. But the point is it's hit and miss. Then even if you're near a Turing island, this clump of cell wouldn't know that there's a Turing island nearby. So now the scenario might be slightly different now if you have everything coupled to limit cycles, to oscillators. So each of these concentrations is oscillating and they're weakly coupled. So now in this high dimensional parameter space, these parameters actually go in these limit cycles. And so the question is now, is it more likely for some average cell clump to actually identify discover a Turing island? So maybe it's less hit and miss and so on. And if that's the case, then maybe, fast forward a couple billion years, and this cycle might turn into a developmental cycle, let's say of an embryo. So there's a Turing island here to form the body plan. Maybe before there's a sort of French flag domain, which is stabilizing Turing or aligns it. And there's other stuff. So that's sort of the idea. So does it help having Turing limit cycles? So here, so this is work done by Brandon. So what he did is he picked a two-equation model, relatively simple. We don't want to make it complicated. Hill functions. And then two of these parameters are coupled to a limit cycle, very simply implemented by these variables, radius and the angle. And there's noise as well, in principle. And so the idea is now, what you see in blue is a Turing island in this two-dimensional parameter space, and now this cycles through it. The good thing is there's already some robustness because wherever you start, inside or outside, it is attracted to the limit cycle, and then it goes through the thing and enters the limit cycle, the Turing island. So there's already this robustness just having this oscillator because it's attracting initial conditions. Okay, and so how do we quantify now the pattern quality and what we see here? So this is sort of shown on the right side. So this is shown here. So when you go to this Turing island, you see, there's some quantity we call SEF. So that describes how good the pattern is, and it goes up, and then it vanishes outside. And this is sort of shown here. So what quantity do we use? We want a simple scalar metric. So we use Shannon entropy, which describes how localized or structured something is a distribution, but we do this in Fourier space. So the idea is now, if you have a nice pattern or a peak in real space, the Fourier transformation of something sharp in real space is very broad. Here on a ring, here very broad. And so this is high entropy, but that corresponds to a good pattern. And so a higher value means a better pattern. And here's some exploration in terms of conditions. So that's a good measure, fulfills certain criteria. And so what Brandon here did is, he compared different ways how to discover these Turing islands. So here, random sampling of parameter space, a bit like in the schematic I showed you. He limited to a circle. In the middle, there's an artificial little island, Turing island. And so the question is, often does it hit it? And here, circles and two types of ovals. How does it compare to identify Turing islands? And here, that's essentially the result. So one thing is the encounter probability measured. So it's basically the fraction of points which are within the Turing island. And overall, this is very similar amongst the different approaches, also random sampling. Of course, it decays with the domain size, yeah, because the Turing island gets effectively smaller and smaller. And so there's not much difference in that way. But what, if it's important for the clump of cells and so on, the organism later, to find it at least, to intersect it. So it's more like a yes/no question. Does it intersect or not by a limit cycle? And this probability is very high, at least for small domains, and decays much more slowly. And so basically, quite deterministically, or almost deterministically, the limit cycle allows the system to run through the Turing island. And because it's done repeatedly, you could say then evolution can act on that and tinker and improve it further in terms of robustness. So yeah, so that was again, hit and miss here, and this is more deterministic. Okay, so here is what is basically shown here is that the shape of the Turing islands matters for the discovery. So here, essentially, what is shown here is a sort of a elongated, bifurcated Turing island. What is shown in gray is sort of an average Turing island, and here on the right you have more circular ones. And then basically, you ask essentially for limit cycles which have a high probability of intersecting, how is their circularity? And for the high intersection probability, the circularity is very low. Yeah, so these are the ellipses. And for the weak intersection probability, it's more circular. So it makes sense, if you have an elongated Turing island and your ellipse is also very elongated. It's a bit like two parallel lines. Of course, they don't intersect, but generally things are not perfectly parallel, and then you have intersection between these two objects. Okay, so the question is then, what happens when you add noise to these limit cycles? So radial and tangential noise. And let's look at these 10 different scenarios. So all of them intersect. So that's somewhat artificial, but we look at variability for these different scenarios. And so what is happening here now with the limit cycles, this index here, this metric shows, of course, huge variability, but overall there's qualitatively very similar behavior. So it goes through a Turing island, and then it gets out of it and goes in again. So similar curves. I should also mention, here we never talk really about steady state because you enter Turing island, there's some delay, it doesn't form immediately the Turing pattern, and when it leaves the Turing island, the pattern lingers a bit and so on. So there's qualitatively similar behavior overall. But then if you do the random sampling for these 10 different circles, you see these bimodal distributions, so, very large variability. So small quality of pattern outside, and then here's a broader peak describing inside, the pattern changes once you're inside, depending where you are exactly. So and then we can also look at standard deviation across these different 10 circles. And so the standard deviation of this metric, so it's a bit interesting because it's the standard deviation itself is a distribution. So the standard deviation is because we're going in a circle and the different values and comparing these. And then there's also noise. So that's why there's a distribution of the distribution. But if you compare this with the other one, the random sampling, this is roughly half the variability. Yeah. So there's also that added value as well. Okay, so let's fast forward basically by evolution, a couple billion years, and now let's talk about the embryonic development cycle. So what if you now have also a French flag, a gradient in the basal expression? So one of the parameters has a gradient, and now let's compare different embryos basically, there's of course some variability and noise, and see how reproducible those embryos are. So you see now as a function of time, this index is lower with a gradient, which is a bit surprising. So meaning less good patterns with a gradient, but it's clear because some of the parameters values don't produce patterns at all. So that's understandable. But then when you look at the pixel-wise comparison, the error is much smaller, the pixel-wise deviations, than without a gradient. So there's more reproducibility, even though there are less patterns. And so let's look in this sort of interval here, what happens at different time points. So this is basically shown here. So here you see without a gradient, you get nice patterns everywhere, but they are not reproducible. These are more reproducible, even so you don't have pattern everywhere. And here's the standard deviation. So larger variability, less variability. So that's so you have more reproducibility of things. So limit cycles identify, discover patterns more easily, and it's also more robust by attracting to a limit cycle and less variability. And it might be useful for pattern formation embryogenesis. So okay, I'm realizing I speak slower than anticipated. So let's briefly talk about this other thing, this other project about sort of the inverse problem that's done by Sichen, and it's still ongoing. So before I talked about the inverse problem, identifying parameters given a model and a pattern. Here it's a bit more challenging now. We are saying there's a pattern, but we don't know the model. And it's about discovering that model. And ideally a very robust model. And this is based on a method developed by Tom Aldrich at Imperial College, and it's called recurrent neural chemical reaction networks. So Sichen, what he did is he basically then extended this to spatial Turing patterns that worked, and sort of ongoing work is extending it to gene regulatory networks. So just in a nutshell, yeah. So without going into too much detail, so the idea is you basically train a neural network to produce a pattern, and the neural network, once it's trained, is producing your chemical reaction network or gene regulatory network. So you get the model out after training. So that's the main essence. And so in principle, in the original model, you have an executive species and a chemical perceptron species, and there's a separation of timescales. The first one is slow, the other one is fast, and they catalyze each other's expression, production, and degradation. These are on purpose very simple reactions because Tom was interested in DNA strand displacement reactions. So here is ODEs, mass action. You can see it more nicely. Y catalyzes the degradation of X. X catalyzes the production of Y. And it's nice and simple. That can be extended to many species. That's an index I and J, not unsurprisingly. And the cool thing now is, why going through all these exercises, the fast species you can think of as quasi-steady state, and then this expression of quasi-steady state actually looks like a smoothed ReLU function, which is a typical activation function in deep learning. It describes the input-output relationship of a perceptron. Yeah, that's why it's called perceptron species. And so essentially that maps a chemical reaction network onto a Turing, onto a chemical reaction network. So the idea is now in the first step of this algorithm, you assume quasi-steady state. That makes it look like a neural network. You can approximate essentially anything, train it by backpropagation. In this case, originally it was applied to dynamical systems only. So it was fitted to train towards a flow diagram. So it makes it also very robust because you can start anywhere and it behaves nicely. So it makes it independent of initial conditions. And then you try to see if it works for finite reaction dynamics because in reality things are finite and it should also work. And that was proved by perturbation theory. And so what Sichen did is now he extended this to spatial pattern. This is a Turing model effectively. So now you have to train diffusion as well. The problem is you can't train to flow fields anymore because there's spatial dependence. So essentially it's trained on movies or snapshots of the pattern formation. And this is basically an outcome looking at the simple Gira-Minot model here. And you can see here now, this is the target which is used for training. And then this is the chemical reaction network, which is trained and then run. And it can actually looks almost identical. There's a little bump here in the arrow between target and the output, but the dynamics are a bit off, but the final pattern is perfect. And so that was a stripe pattern. Now, if you go to a spot pattern in this case, for different parameter combinations, it doesn't work so well. There are larger errors, but it's again, the dynamics are messed up, not the actual pattern, which looks almost identical here. Here's a bit of investigation of, what happens if you now change for your final simulation on the slower time scale. If you change the initial condition, initial noise is different. So it still works. The error between the target and so on, the final output is very similar. The patterns are very similar. Also, the linear stability analysis, interestingly, the dispersion relationship matches as well, because it's not unsurprisingly to some degree, because it forms a pattern. And it doesn't look perfectly like the same, but the peak is important for the instability, and that looks very similar. And the key thing was, the problem was with the original Turing is the two-equation Turing is that the diffusion constant have to be very different for activating inhibitor. Here, Chen said, okay, let's make them more similar and see if the training can still work, if it can compensate for that. Yes, the training loss gets larger going away from this ratio. And yes, the error between output and input get larger. But overall, the patterns look nearly perfect. And so now ongoing work is essentially how to change the chemical reaction network to something which is more important for synthetic biology, in particular gene regulatory networks. And so the idea is now to go away from ReLU to something which looks like a sigmoid function, but actually Hill functions. So you can imagine it's a bit harder because there's these vanishing gradient problems and so on. But so that's the idea. So just a little flavor. I mean, I'm almost done here. So the executives, the slow species would be the proteins, and the fast species might just be the promoters with fast binding and unbinding. And they can be implement this differently in different ways. Here in the simple case, it's competitive binding, so they're all competing for the same promoter and inhibitor and activator. And then here with the finite binding dynamics. So the thing is now, that didn't work for Turing yet, surprisingly, but it worked at least for the repressilator. So Zichen here tried to, so you have some oscillations of the repressilator model and training it on that worked. Interestingly, which is shown here on the right, interestingly, the pathway looks a tiny bit different. So you can see already how this is good for model discovery. So the red arrows are inhibition. One inhibits two, inhibits three, inhibits one, like here. But you also have positive feedback going backward and self-activation. So that's interesting. So this is promising, but Turing hasn't worked it. Okay, so the original approach was chemical reactions, simple second order mass action kinetics, robust initial conditions, worked even for Turing, and now with the gene regulatory one, it's much harder. It's not fully clear. It's probably of the vanishing gradients, which makes it training harder. But there are different ways how to implement it, so we are playing around with this. Okay, so that's essentially the summary. The topics I talked about, starting with synthetic biology and brute force mathematical modeling with very complicated models, and also then using pins to parameter inference on simpler numerical patterns and chemical patterns. And in the middle part, I talked about how to discover these parameter spaces, these small Turing islands by limit cycles, which are abundant in nature. Everything is essentially oscillating. And then in the very last part, I talked about these recurrent neurochemical reaction networks and how to extend them to gene regulatory networks to discover basically also the models, the Turing topologies. And so this is ongoing. So, okay, so I finish here with some acknowledgements. So on the experimental side, there's Mark Iserland's lab. A lot of the work was done by Yuri Ticker, but also George Wachter, PhD student. And on the theory side, there was Antonio, a former PhD student. He did the pins. So Brandon did he was an undergraduate. He's now a master's student at Imperial College. He did the limit cycles. Zichen, also an undergraduate, is now a PhD student in Cambridge. He did the chemical reaction networks. And Martina and Rusbe were involved in the initial part modeling the synthetic biology experiments. So I think I stop here, and yeah, I'm happy to answer any questions you might have.
Okay, thank you so much, Professor Endres. If there are any questions, you could just unmute yourself and yeah.
Yeah, Daniel. Yeah.
Uh, David?
Yes. I don't suppose this is at all relevant, but I have spent my life trying to persuade people that Hill functions can't be derived from the law of mass action. They're a sort of extreme case of infinite interaction energy or something in the case of an ion channel.
Hmm.
So, but I guess they are sufficiently flexible in their shape that they'll suffice in your case, although they're not physically realistic.
Yeah, I mean, interesting question. So, I mean, it's basically, allosteric interaction, so cooperativity, so transcription factors binding to the promoters and, you know, uh, having one bound makes it easier for the other one because of allosteric interactions. So you could build something like an MWC model, Monod-Weyman-Changeux, which is, I think, based on elementary reactions actually. And then you can,
Yeah.
derive in certain limits, essentially a Hill function. A Hill function is basically the limit where you have all or none binding. Yes, it's a bit artificial, but you can get something similar from a set of mass action kinetics, which, yeah.
Well, not that similar. I mean, hemoglobin has four subunits, but the Hill coefficient is only about 2.4 or something, not four. Better you can mimic it. Yeah. Thank you.
Yeah, yeah, I mean, I thought, because of the cooperativity of four subunits, you get closer to four. I thought it's 3.6 for hemoglobin. But anyway, I mean,
No,
we need
no.
a certain degree of nonlinearity, and a dimer formation with a Hill coefficient of 2 suffices in most conditions. So we might not need very high Hill coefficients, which are hard to achieve.
Yeah.
Yeah, thank you.
Thank you.
Thank you. Any other questions? So I may take this chance to ask a question. So for the physically informed neural network, if you try to model, because I'm not really familiar with this, if you try to model kind of the reaction networks with PIN, um, can we kind of recover the mechanistic model parameters from the neural network, or it's like try to model it with totally a different system so we cannot get all these physical insights?
Yeah, I mean, yeah, good question. So the idea is, to have in the loss functions the so-called PDE loss. So we don't want to only describe the pattern with many, many parameters of the neural network. We also want to make sure whatever pattern you get in your approximation is actually fulfilling the differential equations or the PDE, sorry. And so you do get the model parameters out. And so the question, of course, is, are these model parameters, similar to the numerical ones from the numerical pattern you put in your simple benchmarking approach. Ultimately, you want an experimental pattern, of course. And so that's what was one of our metrics, how close are these parameters to the original ones produced for the original pattern? And as I showed in this graph, so without noise, it's perfectly reproduced. And this noise, things degrade and it goes up the error. And but so that works quite well. I mean, there's more to be said. It's sensitive in one hand, but also it can be rescued with more data, sort of law of large numbers. And that was for least squares, and then the pins is more robust. And parameters might not match perfectly, but whatever pattern you get out looks similar. It's not perfect. It looks similar, and the parameters are not perfect, but it's very robust to additive noise. I don't know if you answered your question. So the idea is to get the parameters out which are meaningful. And for pin,
Okay,
it's a bit
yeah.
more flexible, but for the other method, least squares, it's better but more sensitive to noise somehow.
Okay. So yeah, thank you for the insights. That's really
So
important and
just
answers
to
my
add,
question.
the idea is to make neural networks interpretable, and that's the pins are a nice way to do it. Yeah.
Okay. Thank you. Any more questions from the audience? If no more questions, let's thank Professor Endres again for his nice talk, and we'll stop recording and put it on the website. And yeah, we hope to see you 1 month later